Well, thank you very much, Dr. Froschper, and to the Vice Chancellor for that very kind introduction. And let me begin by thanking the Nobel Committee and the Nobel Assembly on behalf of myself and my colleagues, the Mosers, for deeming our research worthy of this very prestigious prize. I think there's no doubt that the Nobel Prize is the most prestigious prize that you can earn in medicine or physiology. And let me also thank the committee and the Karolinska Institute for inviting us here to give these lectures. So my lecture today is on the spatial cells in the hippocampal formation. And the intention is to take you right back to the beginnings and give you some idea of what we were facing when we first started looking at these cells, where we were drawing our inspirations from, and to also give you some idea of the methods and the way in which we study these cells, and also some of the things we know about these cells. We can't, obviously, in a short lecture like this, tell you everything. There's actually a fairly large industry out there now studying these cells. But I'll try to give you some idea of what we know about them. Now. So my introduction to the hippocampus came when I was a PhD student at McGill, where, as Professor Froschper says, I was a student of Ronald Melzack, and I was lucky enough to have very influential and tremendously generous mentors, particularly Ken Casey and Herman Baumer, who helped me develop many of the techniques which we eventually used to study the hippocampus. At McGill, we were introduced. Let me see if does that work? That's not working anyway, is it? Okay. We were introduced to the hippocampus by Brenda Milner. And she told us that it was a memory system. And she told us this on the basis of her studies of, as he was known then, HM, as he is now known since his death, Henry Morrison, who was unfortunate to have acquired epilepsy at an early stage. It was severe epilepsy, which wasn't controlled by anti-epileptic drugs, and for which he was elected radical surgery, which was the removal of structures in the mesial temporal lobes here. This is the intact hippocampal region here, and this is the removal in HM. And following the surgery, HM had amelioration of his epileptic fits, but on the other hand, he had acquired a very dense and severe amnesia. He no longer could remember the events of his day-to-day life, and he could no longer remember events that had happened in the past. And after many, many years of study of HM and similar patients, particularly by Suzanne Corkin, who was a student of Brenda Milner, they concluded that the major deficit in HM was that he cannot recall anything that relied on personal experience, what we call autobiographical memory, such as a specific Christmas gift his father had given him. He retained only the gist of personally experienced events, plain facts, but no recollection of specific episodes. He had what we now describe as an episodic memory deficit. So let's just look a little bit about the structures that were removed in HM. Here's the hippocampus in the human, and here it is in this very nice revolving three-dimensional display. And you can see it sits right inside the temporal lobes, quite buried deep in the temporal lobes, far away from either the sensory inputs or the motor outputs. If we do a cross-section through it, then we see this very nice, in this multicolored stain section here, this very nice laminated structure where all the cell bodies are lined up right next to each other in this beautiful, beautiful arrangement, which is very much beloved of anatomists and physiologists. I won't go very much into anatomy, but we can talk about three major subcomponents of the hippocampal formation and the related structures. We can talk about the hippocampus proper here, with the CA fields and the dentate. We can talk about the entorhinal cortex, the area that much beloved by the Mosers, my colleagues, and where they found the grid cells, and the intermediate structures in the cerviculum here. And I'll try to tell you a little bit about what the spatial cells in these different regions are doing. So when we first started approaching the hippocampus, mindful of the fact that we were probably looking at a memory system, we decided to place probes, microelectrodes, into this region here in the CA1 field of the hippocampus, and to see if we could see what the cells were interested in, and particularly if they were interested in particular kinds of memory. And I must say, I was pretty Catholic at that stage, Catholic with a small c. I was prepared to see that the cells would fire to all sorts of memories, all sorts of stimuli. Instead, what we found is shown in this picture from our first paper here in 1971, is that the cells were only interested in a very specific type of behavior that the animal was engaged in. They weren't, in fact, interested in most types of memories. They weren't, in fact, interested in most types of behaviors. They were only interested in where the animal was in the environment. The cells were coding for some aspect of space. They didn't care why the animal went there. They didn't care what the animal did there, by and large. It was really where the animal went. And you could see here from this very first picture here where the spiking occurs only sometimes here, not here, and not here. And that's only, as shown in this histogram, when the animal goes over to this location here. So, not being of a very conservative bent, I immediately thought, wow, this looks like the type of system that E.C. Tolman, who has already been mentioned, would have loved to believe existed in the rat brain. And what we said was, these findings suggest that the hippocampus provides the rest of the brain with a spatial reference map. And if you didn't have this map, if the animal didn't have this map, it couldn't learn to go from where it happened to be in the environment to a particular place independently of any particular route. It didn't have a flexible representation to produce flexible behavior, as it was might be expected from Tolman's experiments. So who's this Tolman? As you've already heard, Tolman was a very, very important psychologist working on the west coast of America, who in the 1930s and 1940s believed, on the basis of his behavioral experiments on rats running in mazes, that the way in which they solve a maze problem was by employing what he called a cognitive map. And here's a quote from him. "We believe that in the course of learning, and particularly learning mazes, something like a field map of the environment gets established in the rat's brain. The stimuli are worked over into a tentative, cognitive-like map of the environment. And it is this map, including routes and paths and environmental relationships, which finally determine what responses the animal will finally release." So that was the idea. We had to put a little bit more bones, a little bit more flesh on this idea, and we suggested that essentially what you needed for a map was to have a set of place representations of the sort that we had already found, but that that wouldn't be sufficient to have a map. You need to not only know where you are in an environment, what the places, where the places are, but the relationship between the places. So we suggested something of the sort that's shown in this cartoon, that places might be related to each other by the distances and the directions between them. Essentially a set of vectors, so that when an animal was in A, it would know how to get to B by following the vector AB, which represented the direction and the distance between them. We also suggested that you could use this system, for example, to identify a location B, so that if an animal knew that it was in A and it was heading along AB that would call up the representation of B independently of the information, the sensory stimuli that were arriving at location B. So it was a very flexible way of representing the environment, and it was a very flexible way of allowing the animal to navigate in a familiar environment. Around this time, Lynn Adel, who's in the audience someplace, very grateful, and I myself, and this is taken from the mid-70s, began to think more widely about how this could be used and to see if we could apply these ideas to the existing literature about the hippocampus, not only the anatomy and physiology that was known then, but also about whether we could explain the deficits that were produced by damage to the hippocampus. Would a hippocampal rat be able to navigate? Was there evidence in the literature? We were quite taken by the idea that's already been expressed that space is a fundamental aspect of the mind, of the human mind, or even of the animal mind. And we waxed lyrical in the introduction to our book. I've put it in a poetry-like fashion here. We said space plays a part in all our behavior. We live in it, move through it, explore it, defend it. We find it easy enough to point to bits of it. The room, the mantle of the heavens, the gap between two fingers, the place left behind when the piano finally gets moved. We made predictions. We predicted that there would be other signals in the hippocampus, or the broader hippocampal formation, which represented those vectors, directions, distance, and speed of movement. And we also made predictions about the kind of deficits you would see following damage to the hippocampus. And specifically, we said animals shouldn't be able to do place learning. They shouldn't be able to learn about places in the environment. They shouldn't be able to navigate, and particularly navigate flexibly, so that if they learned to go by one route and that route were blocked, they wouldn't be able to go by another route, and they wouldn't have exploration. Well, if you make predictions in science, and you're lucky enough to have good colleagues, they'll test them for you. And the first one to test these predictions was Richard Morris, shown here. And Richard said, well, if you really believe in this idea, let's come up with a task where the animal has to flexibly navigate from different locations to one location in the environment. And he came up with what's now called the Morris water maze. And it's a very simple behavioral task. It's a tank of water filled with cloudy water so that the animal can't see what's underneath the surface of the water, and in which is located a sunken, hidden platform. And the animal's task is very simple. What it has to do when placed down in any location in that bath is to find its way to this sunken platform. Very simple. When they first placed into this swimming maze, the animal wanders around until he eventually bumps into the platform and climbs on it. After a very small number of trials, here's 8 trials, the normal animals go very, very straight, form a very straight trajectory to the goal. And they do that from any location in the water maze. If you now inflict hippocampal damage on the animal, what you find is that whereas normal controls and control animals go straight to the platform, these animals look very much like they did at the early stages. They wander around, can't find the platform until they bump into it. So very good evidence that at least one of the suggestions of the theory was in fact correct. And I have to say that the water maze is still the most popular and most powerful test of hippocampal function that exists today. So what about the cells? Well, one of the predictions was that there would be some representation of speed of movement in the hippocampal formation. Because, of course, if an animal is moving around an environment at different speeds, it has to take its own speed of movement into account and essentially offset it so that if it moves from one place to another rapidly, the brain will be able to take that into account as well as if it moves from one place to another slowly. And we were very fortunate to have Bruce McNaughton, and Carol Barnes come to our lab as postdocs in the early 80s. And one of the things that they showed was that the firing of the place cells as an animal ran around the environment actually changed as a function of how fast the animal was running. And you can see here that as the animal ran faster, the firing of the cells increased. And we also have subsequently been able to show that there are specialized cells, we haven't found very many of them, but specialized cells in the hippocampal formation which look for all purposes as though they are, in fact, tracking the animal's speed. The faster the animal runs, as shown here from 0 to 1 meter, the higher the frequency of these cells. What about the other cells? Well, we now know through the work of many laboratories, including, as you'll hear subsequently, the work of the Mosers, that there are several other kinds of spatial cells in the hippocampus. And together, we think that we have all of the elements to form a cognitive map. So one of the things I'll do is I'll just give you a very quick run-through of the different kinds of spatial cells that have been found in our laboratory and in other people's laboratory. And briefly, what they are is they're place cells, which represent where the animal is. They're boundary cells, which tell the animal how far it is from landscapes, and particularly walls in the environment. There are head direction cells, which tell the animal what direction it's pointing in an environment. And finally, there are the grid cells, which I'll say very little about because I'll leave it to the Moses to describe them in much, much greater detail. So let's go through and look at these cells in a bit more detail. So first, there are the place cells. If we place an animal, this is a cartoon made by my colleague Julia Krupich. If we place an animal into a circular environment, we watch where it is using an overhead camera, and we also register where it is when one of these place cells fires. What we see is, by and large, the cell is silent over most of the environment, and it's only when the animal goes into the part of the environment which is preferred by that cell, its place field, that the cell fires. And so you can see all of these red dots are adding up. And what we typically use is a heat map to represent the firing rate of the cells. So the heat map shows that this cell fires with a maximal firing rate of 7 hertz here, and successively lower rates as you move away from the center. And this is a typical type of way in which we represent these cells. If we look, and one thing that I should point out is that if an animal runs around in an environment like this in an open field, it turns out that it doesn't make any difference which direction the animal runs through the place field. If it runs north, the cell is happy to fire. If it runs south, it's equally happy to fire. It looks as though the cell is representing the location and not something about the sensory impressions that the animal is getting in that location. Well, if we record from not one cell, but many cells at the same time, we see what is now the standard story about these cells, which is that each cell has its own preferred location. Different cells have different preferred locations. And if you take about 30 some odd cells recorded at the same time, you find that the entire environment is mapped. There is always a cell firing whenever the animal is in a particular location. So it looks as though you have the information about all of the locations in an environment, and you can imagine that what you're doing is looking at the neural substrate of a mapping structure. The same cells are used over again. So if you record from the same cells in 2 environments, you find the following kind of pattern of activity. Here we're recording from animals in a circle and a square, and you see that there are some cells which only fire in the circle and not in the square. There are other cells which only fire in the circle and not fire in the square and not in the circle. And there are some cells which fire in both, but in different locations. So it looks as though you need to look at a pattern of activity across a group of cells to actually know where the animal is. Now, over the years, we've always wondered whether these were really 2-dimensional representations or whether they were actually 3-dimensional representations. And if you want to actually study a 3-dimensional representation, then you should study them in an animal which is happy to move or usually moves in 3 dimensions. And that's what Ulanovsky and his colleagues have done in the bat. So they've recorded, technically very difficult, they've managed to record from these cells in flying bats. And what you can see here is that each of these cells is now not a two-dimensional dual Gaussian or double Gaussian structure, but in fact they look like they're three-dimensional spheres. And if you record from a group of them, as shown here, say 10 of them at the same time, what you see is, again, the 10 cells essentially tessellate now the volume of space and represent the entire location. So we know that these cells, at least in bats, and we're pretty sure in rats as well, are three-dimensional representations. Now, one thing about these cells is that they are, in fact, really quite clever, cunning creatures. One of the things I haven't mentioned is that if you look at the gross activity, the LFP as we call it, activity in the hippocampus, which seems to be representing the activity of large numbers of cells at the same time, you see something rather striking in terms of brain research. And that is that sometimes this LFP activity takes on a very nice sinusoidal rhythmic activity. And when people first saw this, they thought, nah, that can't be right. The brain doesn't give out sine waves. But in fact, it is a real activity. And as I'll show you in a second, it actually is a very, very important aspect of hippocampal function. Over the years, so when the animal is sitting quietly, then you get this rather irregular activity. But when the animal starts running around, they're shown here, then you get this very nice sinusoidal activity, which has a frequency of about 8 to 10 hertz in the rat. Over the years, Yuri Buzaki, who has spent his life studying this and other rhythms in the hippocampus, would meet me in various venues and say, come on, John, what's the role of the theta activity, as it's called, in hippocampal function? What are the place cells doing when the animal is in theta activity? And I assured him for many years that there was no relationship between them. But somewhere in the 1990s, Michael Recchi and I decided we'd better look carefully. And we saw something very interesting, and I think of general significance for not only the hippocampus, but other kinds of coding in the nervous system. And this is that the cells, in addition to using the rate of firing of the cells in the hippocampus, also use the timing of the spikes. They use the actual time at which the cells fire to provide information to other parts of the brain. And that's shown in this figure here, although I don't think you'll be able to see it actually. If we record as an animal is running along around an environment and look at when the cells fire, they don't actually always stay locked to the same part of the theta wave. They move around. And they move around in a very systematic way. And if you ask how that systematic movement it maps onto the environment of the actual location in the environment, what you see is there's a very systematic relationship here. So a cell which fires here will show a very systematic change in its reference and relationship to the theta waves, suggesting that position is coded not only by the rate of firing of the cell, but also by the exact timing of the cell. The time at which the cells fire relative to this wave are actually conveying information. And that's probably a very important and general property of cells in the cortex. Okay, where does the information about the environment come from? So we know that animals can tell where they are on the basis of where they are, on the basis of sensory inputs coming from the environment. And the first clue about that came from an experiment that Neil Burgess and I did, in which we recorded from these place cells in boxes with different ratios between the sides of the box. So we recorded in squares, small squares, large squares, and in rectangles, which had one side representing this, one of the squares and the other side of the size of the other square. So we essentially were taking squares and stretching them out into rectangles. What would this do to the way in which the cells were firing? And what we saw is that the cells were stretched out quite often, so that the fields stretched along the distance that we were stretching the box. The cells seemed to be trying to represent the distance from each of the walls. So you can imagine that this cell is trying to stay close to firing next to this wall, but also close to, say, next to this wall. So that as you pull the walls apart, the cell is trying to stay next to them and get stretched in that direction. Well, I won't go into it, but we suggested that maybe the inputs to the place cell, or at least one of the inputs to the place cells, was actually of the sort of representation that you see in these Rothko paintings. And of course, what Rothko is famous for is painting simple paintings in which there is a single stripe at a particular distance from one edge of the painting. And you can imagine that the brain could use this kind of information to tell the animal, you are now a certain distance from a particular edge of the environment, a particular wall in the environment. And you can imagine if you could put a couple of these together, the overlap between them might actually represent where the animal's exact location is. And that's what this little bit of mathematical computation here suggests. Well, you have to be careful again what you predict, because following that prediction, Colin Lever in our lab went looking for these cells, and he looked not in the hippocampus, but in a neighboring area, the entorhinal, the subiculum. And about the same time, Solstad in the Moses lab was looking in the entorhinal cortex and seeing rather similar things. And what they both saw is that there are, in fact, cells which have this Rothko-like stripy representation. Some of them are close to one wall, some to another wall, some are vertical, some are horizontal. And if you put these together, you can see that if you just take these two and overlay them, what they will produce is, in fact, a place-like representation. So we now think that the information about where the animal is coming in from the environment is represented by these kinds of what we call boundary cells and what the Moses call border cells. Well, how about the information about directions? It took until about 1984, 1985 before these cells were actually identified by Jim Rong. Here's Jim and his colleagues Jeff Taube and Bob Muller, who did most of the early work on these cells. They're called head direction cells. Unfortunately, Bob Muller died 2 years ago, and he's a great loss to the field. But I thought I would just show you what one of these cells looks like. So here's an animal again. We're watching where the cells fire. And every time the cell fires, we represent the animal's head and direction of pointing. So as you see, as it goes around, it drops these arrows here. And at the same time, there's a summed arrow representing the overall representation of distance. And what you can see is that most of these arrows are pointing in the north or slightly northwest direction. The cells are representing the animal's heading within the environment, irrespective of where it is in the environment. They're as happy to fire here as they are there. These cells are found in an area just between the entorhinal cortex here and the hippocampus, and they're called head direction cells. One of the interesting things about these cells is that if you look at a couple of them, they seem to be tied together. Their fates are bound together in a virtually ineluctable fashion. If you have a cell which is representing a north direction here, and two other ones which have other directions, and then you manipulate something about the environment to get the cell to fire in the east direction here, you find that the other cells modify their preferred directions to maintain the same angles between them. It looks as though this whole system is wired together to represent a polar coordinate system which the animal can use wherever it is in the environment to tell the direction of heading. Okay, well, what about how distance is measured? In addition to direction, we need to have a distance metric. Well, is distance in the brain represented as William Blake would have here in his picture of Newton by calipers, which are then used to lay the difference between 2 points along a one-dimensional ruler? Or is it something more complicated, shown by the Liechtenstein yellow cliff painting here, which is to sort of have a mesh-like representation, a two-dimensional representation, where you could tell, for example, how far up the cliffs you were by counting the number of these dots, these Bengo dots, or how far in you were by the same technique. Well, it's clear from the work of the Moses, and you'll hear a lot more about that in the next 2 lectures, that it's the latter, representation. Because what they found in the entorhinal cortex are a set of grid cells which look very much like those Bengay dots. They represent the environment as a series of firing fields which are laid out in this beautiful, symmetrical, hexagonal pattern, and which is reproducible from one trial to the next, and which in most environments can be used to represent distances in the environment. I won't say much about these. Edvard and May-Britt will tell you more about them. You can characterize each of these patterns by the distance between the 2 peaks, by the angle of the representation relative to the walls, and the distance from the walls. They'll tell you a lot more about that. The only thing I need to tell you is that they come in different flavors, and particularly for our talk today, they come in different sizes, so that different cells, and particularly cells in different parts of the entorhinal cortex, have different spacing between them. And that's important because if you take cells, 2 grid cells with different spacings, and you simply add them together and do a little bit of mathematical computation, you can also produce a place cell. So we have 2 ways of producing place cells. Thinking about the grid cells, my colleagues Neil Burgess, Caswell Barry, and I began to think that at least one possible way, and I won't tell you how we came to this conclusion, one possible way of producing a grid structure is by overlaying different striped patterns. And you can see this from another Liechtenstein painting here, where he uses these dots again, but he juxtaposes them to these stripes. And if you look at the pattern here, what you see is that each of these patterns of dots here actually is the extension of a stripe running down in this direction. And you can imagine very easily that if you had stripes at different orientations, say 60 degrees relative to each other, that would effectively produce this grid pattern. And this is one of the theories that we and others are working on about how you produce the grid pattern. Well, again, you have to be careful about what you predict, because following that prediction, one of our colleagues, Julia Krupic, actually went and looked for these cells and found, in fact, cells which look very much like the stripy cells which were predicted by this theory. And you can see here that these cells, they're not complete stripes, but they have this very strong striped orientation. So we think there's at least some evidence for the idea that one way you could produce grid cells is by actually overlaying these stripes. So we have then, on the basis of a lot of work in many, many laboratories, we have then a whole panoply of different kinds of spatial cells. We have the original place cells, we have these boundary cells, we have the head direction cells, and the grid cells. And we're still working on mathematical models, as are many, many other people in the field, and there are still many, many questions that we need to answer about the exact way in which these cells are composed. I want to tell you about 2 other aspects of our work before I give over to the Moses. Professor Froschber actually mentioned Immanuel Kant, and I'm a great fan of Immanuel Kant. I have done more than my fair share of philosophical reading and thinking, and I've always been taken by Kant's idea, and it was one of the reasons I was really quite pleased to begin to study space and see these spatial representations. I've been taken by his idea that space was one of the fundamental aspects of the human mind. And what Kant said was that space is nothing but the form of all appearances of outer sense. If you want to think of something that's outside your body, if you want to think of something outside of your mind, then you really need to think of it as located in some space. And it can be given prior to all actual perceptions, and therefore exists in the mind a priori. It can be prior to all experience. He had this idea that you bring, or the mind comes to the world with some machinery, some cognitive machinery that it uses to make sense of the information coming from the world. And one of those was space, another was time. So thinking about this, Nadel and I said, well, yeah, we accept that the representation we're looking at is a three-dimensional Euclidean space, and particularly that this is a spatial and cognitive structure which is a prerequisite to the experience of objects and their emotions. This translates in modern neuroscience terms into the idea that maybe the spatial representation in the hippocampus is there before the animal has had any significant experience with the environment. Many people in the field will think that the way you build up cognitive representations is by taking experiences that you've had and do some manipulation on them, compare them, contrast them, and so on and so forth. But it's possible that at least part of our cognitive representation, and particularly the representation of space, is actually given a priori. And so recently, my colleagues and I have followed this idea, and particularly when I'm talking about Francesca Kikuchi, Tom Wills, and Wei-Min Tan, and tried to see if we could see when these representations became active in young animals as they were developing. And we looked particularly at the early first couple of weeks in life to see when each of the representations appeared, and if we could see if some of them appeared before others, and if some of them appeared before the animal had had much experience of the world. And what we found, to make a long story short, is that the head direction cells are the ones which actually appear earliest. They appear first. They come before the play cells, and they come before the grid cells. So the order is head direction, play cells, and grid cells. Interestingly enough, the directional cells, the head direction cells, actually occur about the time that the animal's eyes are just opening and that the animal is just leaving the nest, i.e. before it's had any substantial sensory experience of the environment. So we think that there's at least some merit in this argument and this idea that part of the spatial system is laid down genetically or epigenetically, but certainly before and perhaps independent of any experience that the animal has had. I think I'm going to do, how am I doing for time? Okay, fine. I think I'm going to quickly jump over the next couple of that slide and just go directly to the implications of this for the human hippocampus. Because I think, as I've already told you, the work of Brendan Miller and many other people have told us that the hippocampus of humans is perhaps a spatial system, but has a broader function, an episodic memory function. So over the years, people have always asked us, can you demonstrate that what you're seeing in the rat has some implications for what we should be thinking about the human hippocampus? And for a long time, it was very difficult for us to think about how we would approach this task, partly because it was very difficult to test neuroscientifically the ability of humans to navigate in very large-scale environments. And it was around the middle of the 1990s that we realized that the new games that were being produced for the entertainment of our youngsters, these first-person shoot-'em-up games where you find yourself in an alien world, go around finding the monsters and shoot them up with incredible guns and so forth, really would provide us, if we could get rid of the monsters and the guns and all the other politically incorrect aspects of the environment. I won't tell you what they were. But if we get rid of those, we would have a wonderful platform for studying spatial navigation across large-scale environments in humans. And we could do, for example, we could do imaging on subjects while they were navigating. And it was Neil Burgess who took apart one of these games, and he got rid of most, as I say, the politically incorrect aspects of it. But he left us with an environment which is about 70 by 70 meters, and which it was reasonably easy to learn to navigate around and find your way around in about an hour for most subjects. And here are a couple of screenshots which show you one of the rooms. Here's a billiard table, here's a bar, there's a broken down cinema, here's what's going on in the cinema. I won't tell you what the original film was that was showing in the film, but there's something for every taste. And so you can use this then to ask people to find their way around this environment and see how they navigate using a cognitive representation. So we teamed up with Eleanor Maguire and ran an experiment in which we did just that. We scanned the brains of subjects and found that, in fact, the only two parts of the brain which were activated during cognitive representations, as opposed to the more routine root-like representations where you just go to one corner and turn left, is the hippocampus shown here. And interestingly enough, how much the hippocampus is activated, how much blood flow there is going through the hippocampus, depends on the actual route and success of that particular navigation. So if people are taking a very straight navigation between two locations, as shown here, as opposed to a wandering navigation like this, you got more activation of the hippocampus, as shown in this graph here. Eleanor then, of course, realizing that there were a whole group of natural expert rat navigators in London, i.e. London taxicab drivers, then went on to investigate the London taxicab drivers' hippocampus and showed, in fact, that they were larger. One part of the hippocampus is larger than you find in most people, and larger, for example, than in bus drivers. Interestingly, she also found that the longer you were a taxicab driver, the bigger that part of the hippocampus grew. So it's not as though you're born to be a taxicab driver. It's that the more you do taxicab drivers types of things, exercise your spatial navigational skills, the larger it gets. So I'm finished now. I just want to summarize. Hippocampal formation provides a cognitive map of a familiar environment. And you can use this to identify the animal or our own current location and to navigate from one place to another. The mapping system uses two very independent and we think very different strategies in order to guide the animal from one place to another. One is based on the environmental landmarks, what you see, what you sense in a particular location. The other on what's called a path integration system, which is information about distances traveled in particular directions. And we think a similar system operates in humans. And we think it can be elaborated, particularly by the addition of linear time and other cognitive abilities to produce this episodic or spatio-temporal memory system, which is the hallmark of the amnesic syndrome. And finally, I would like to thank many of the people who've been involved in this adventure. It's been an exciting adventure. I'd like to thank, as you can see, the many talented colleagues that I've had. Starting from our initial observations, we've been able to interest and excite many very, very talented people, and I've named many of them as I've gone on. Of course, you'll hear from two of them in a moment, the Mosers, who are certainly some of the most talented people in the field. I'd also like to thank my family and my wife, Eileen. It's probably obvious that in order to do this kind of work, you really do need very strong support at home. I've been very lucky to be in a world-class institution, UCL, which has given me the freedom and the support to do this work, and I've also been the beneficiary of considerable and continuous funding over the years. I would particularly like to point out the Gatsby Foundation and the Wellcome Trust, who recently have combined to provide UCL with the money to build the Sainsbury Wellcome Center, which is a new center for the study of neural circuits and behavior. We're trying to do for the other parts of the brain what we have done, we think, partly successfully for the hippocampus. So thank you very much for your attention.